Okay, hopefully people will come back. So the structure of DNA was discovered right around 1942. I'm trying to think. 52, sorry, 52, 53. And it is, the credit goes to 4 scientists. But at that time, like I said, for a long, long time, at least till Rosalind Franklin was alive, she never actually got the credit for all the work she did. During that time, there was another scientist. His name was Erwin Chargaff, and he was comparing different species. And what he found was every single species has the same thing, ATGC or AUGC in RNA or ADGC in DNA. And he would take the DNA tissue from different, tissue from different organisms, and he would find the same thing again and again and again. So what he found was that A is equal to T because A pairs with T. So the amount of A is same as T. And the amount of, so the amount of G is same as C. So that's what it is. So I have a problem for you, okay? So in the sense, like, I'm going to ask you a question. So if I tell you in an organism, so this is hypothetical, okay? There is, so A is, or adenine, or in a sample, adenine is found to be 20%. What is, this is like, if you can do this, then you got at least one of the assignment question. What is T, C, and G? How would you find this for me? And if I'm missing something, you should tell me, like, you are missing this. So if I give you just the adenine in a sample, can you find out what is thymine, what is cytosine, and what is guanine in the sample? Okay. 40. Interesting. Well, you can find thymine really quickly. Come on, I expect you to be able to find the thymine. Yeah. Okay, so yeah, I can see some people. Yeah, I really like it when you guys, you're thinking and you're trying to do, you're trying to use your knowledge and ideas to do this. So I really enjoy that part. So I think I got some correct answers in here, but I'm happy to give you some hints. So first of all, I know for the people who didn't get it, not your fault, we have to remember that A plus T plus G plus C should be 100%. So I think people who were able to solve it, they were able to solve it because even though I didn't give it, but they did assume that all of them together makes 100%. Oops, let's go back. And then they also did guess. They were like, okay, if A is 20%, then they did tell that, oh, then T has to be 20% because they kind of pair together. So how much ever A, that much part is T. Now, what about G and C? So all of them should be 100. So AT should be, AT is 40%, which means that the GC will be 60% combined. So which means that half of the G is 30% and half of it, which means C is also 30%. So is this something that is clear to everyone? I put down I'm trying to put down the answer. There's not like a whole lot of place to put answers. That's why I am keeping it here, but I'm ready to move it if you need it. So did everyone completely understand that? Okay, great. So I really hope you did. And maybe we can try one more. Okay, all right. So why don't we go over it one more time? So basically, amount of A and T are same. Whatever is A, same amount is T. So then if A is 25, T is 25. A is 30, T is 30, like that. So we know at least A and T are 20% each. So if A and T are 20, then combined will be 40. Okay, so that is one thing to remember. And then if A and T is 40 and together AT plus GC is 100. So AT, A plus T plus G plus C is equal to 100%, which means that if this one is 40, so let me just circle that for you. If these two are 40, then these two have to be 60 to make it up 100. 40, 60. Now 16, two things, so G and C are equal to each other. So G plus C is 60. So G should be 30 and C should be 30. Okay, that's how you do it. Okay, so let's just clear this one. Okay, so remember this rule, and this is very important. You will see that we are using it a lot. You can see here AT pairs with GC, and that's what it is always. So if you see this example, ATCCGGT, the complementary is A pairs with T, T pairs with A, C pairs with G, G pairs with C, so on and so forth. So you can see that correlation, that A pairs with T, G pairs with C clearly in this example. Now, if you're wondering what is 5' and what is 3' end, that is the numbering of the carbon. So let me just go back here. Let me go back here in this figure. That's why I have the detailed figure for you. So if you remember, we talked about how carbon is numbered. It's always numbered starting on this end, which is typically the anomeric carbon. So this is 1, 2, 3, 4, 5. So 5' is where the DNA starts. And where does it end? Where the 3' is. So 5' to 3'. And that is what, if you see those numbers ever, 5' to 3', that is the reason why it's like that. Okay. So just, you don't need to know that so much, but I mean, if you do not have any missing gaps in learning, then it's a better learning. I mean, you might encounter it elsewhere and wonder what this is. Okay, so if you see this structure, you can clearly see each of the nucleotide in here. You can see those base pairs in here, which is shown over here. And this is angstrom. So angstrom is 10 power minus 10 meter. So that would be 10 to the power, which I'm going to write in parentheses because Zoom doesn't let me write superscript. 10 power minus 10 meters is angstrom. Okay, which is really, really small. So there is a question that I'm going to ask you, but I'm also going to give you the answer right away because I know that a lot of people will tell that an expected answer would be microscope. So if this is so small, micrometer is 10 power minus 6 meter. And what we are talking about distance is 10 power minus 10 meters. So if the if you look at the DNA under the microscope, will you be able to see these nucleotides? Yes or no? Or is it too small and is not observable under microscope? Can you actually see a DNA helix if you use a microscope? What do you think? Do you think DNA is too small to be seen under microscope, or you can see it under the microscope? Like, basically the question is, can you see it under the microscope? So you can just respond in a yes or a no. Okay, I heard from at least one. DNA is really small. You can see the bacteria under the microscope because bacteria is 1 micrometer. But for DNA, typically they are doing either an extremely specialized, not even extremely specialized microscope. It has to be, they do X-ray crystallography to find out the structure of the DNA. So it is by crystallography or very indirect methods. It's not visible under the microscope. So just wanted to let you know. A lot of times, I used to do strawberry DNA extraction. I don't know if you've done that lab. It's very easy, especially if you have kids at home who are like even 4-year-olds. Like I would do the experiment with them. It's really fun. So all you do is you take the strawberry and then you mash it and add some soap and salt water, you can find the protocol really easily. And then you strain the mashed mixture in a tube, and then you are putting some ethanol and um, yeah. So what happens is that essentially you do get the DNA out. And then I asked them, if you see it under the microscope, can you see the double helix? And almost everyone in the class would say yes. But the answer is like, you cannot see it. It's just too small. The dimensions is molecule, and you can never see molecules under the microscope. So just fun stuff, not really related to, maybe dental hygiene directly, but still things that you should definitely know. So these two chains are complementary. You can see the complementary base pairing here. They run anti-parallel. You can see 5 is here on the top, 3 on the bottom. This one, 3 is on the top, 5 is on the bottom, anti-parallel. So you're going in a different direction. The two chains have different sequences a little bit, but they're actually complementary to each other. So if you start reading from here, you can find the complementary sequence easily. And if you see here, this is what it is, CAA. So that's what this is, CAA. And then TCG, TCG, TCA. That's what it is. Okay. And then you can see the complementary base pair is like that. GTTAG, which is right here, GTTAG, and then CAGT. Okay. So just for again some general information in here, the genes are made up of DNA, and human beings have 3.1 billion base pairs of DNA, and they have about 30,000 genes. So the percentage of gene that is actually useful in human is just 1 to 2%. The rest of it is so far called junk DNA, but we do know that it has some purpose. So only 1 to 2% is what makes something useful in your body. So now let's move on from the DNA and get to RNA. So when we are talking about RNA, there is a group of 3 nucleotides of RNA. They are called a codon. So if you see here on this table, which is marked with red and blue, you can clearly see here that there is these UUU listed or UUC listed, and these make one codon. So this is what makes a codon, and this is what makes these amino acid. Okay, so I do have a little bit of homework for you, in the sense like you need to try it right now. Maybe I should call it classwork. Okay, so what you need to do is convert this DNA to RNA using the complementary base pair rules that we just learned, that is A pairs with T and G pairs with C. Then you have another task. So that is conversion of DNA to RNA, and that is called transcription. Then you use that RNA sequence and convert it to amino acid, and that is translation. So if you see on this side, on this table, you can see that AUG. So I'm going to show you how to do this. So let us take a look at this. So AUG. So this is A. So let me probably do this in yellow. So A, U. So wherever A and U meet, so I'm trying to show you A and U. So this box, AUG, AUG is a M, and that's what is written here. So like that, you should also go the next step and find out what is the outcome of transcription as well as translation. So I want to give you a couple of minutes. You can just write the answer in the chat if you wish to, and we'll go over it in a few minutes. Oh, okay. Yeah, that's fine. If you're doing something, then I can totally understand. Yes, exactly. In the bed. Yes, you start wherever it's left. It is started at one point, so you can actually feel comfortable and get kind of going. Oops, I'm sorry. Wow, it won't let me do anything. Okay, I'm sorry about that. Let me. So if were you able to, okay, let's see. We can break this in chunks. So let me try to. So for TTG, were you at least able to find for TTG? Okay, someone wrote. AAC, GGA, yeah, not too bad. Okay, yes, I think I do agree with you. AAC, so that would be, okay, yeah, TTG is AAC. Okay, now the AAC, what is the, okay, let's finish this one, AAC. So now let's do the next one. The next is CCT. So what would be the answer for complementary base for this one? And you can write in the chat or you can just say it. If you are not getting this for any reason, you must ask me. Because I think we don't want you to be, no one to be falling behind this, right? Okay, so yeah, GGA, yes. Okay, so what is the next one after this? So you get the point, like you know, you can just keep going and you can, so we'll do just a couple more. So let us see. Now we will do, get a little bit bold, CCT, TGA, and yeah, let's do two at a time. So see, what is for CTG and what is for TGA? So let's start writing that down. The opposite of C or complementary is G, of T is A, and of G is C. For next one, it is A, and then C. And then what should be the last one? Yes, I'm glad you didn't get tricked. So ACU, like that, okay? So the next one after this is CTC, which will be, now let's go a little bit faster, GAG. Then AGT will be QCA. Then CCC, and then just give me one. Yeah, okay. So if you see here, then this will be CCC, and then we have only two more remaining. So that will be GAA, and the last one is TGA. Oh, good question. What happens if one or two are left? That's a mistake. That's a mistake. You know, all sorts of mistake happen. You are talking about what if one or two are left. I will tell you some real bad scenarios which actually happen in the human body, you know. Okay, so now with this, now you got to do even a bigger task than what we were doing before. So tell me what should come here. And I would really, really want to hear from as many as people that can tell me by looking at that table. I hope I'm not, am I blocking? I think I'm blocking a little bit. So what is the answer for AAC? I saw someone said N. A C. Yes. Yes, my friends, that is perfect. And then what about, okay, so AAC, and then let's try the GGA. Let's find the GGA. Where's GGA? G, and then last G, and then A. Okay, yeah, I found it. I'm waiting for you guys. Perfect. The next one, that is GAC. And okay, now I'm going to give you a bigger task while I'll ask other people if they're understanding it. So the bigger task is, can you find the remaining 1, 2, 3, 4, 5, 6 remaining and write it down? Okay, so for people who understood, they can write down the remaining. If you did not understand this, this is a good time to ask. This is also something that's, of course, something that you are probably going to see in assignment as well as other places, right? Quizzes, exams, and all that. So if any part was confusing, whether transcription or translation, please do let me know. Okay. Okay, so I'm going to go over it one more time. And this time, if for anyone, if you did not quite understand this, you can just do, and for the others, you can just kind of tune me out and just try to find out the amino acid. So the first step is you are given a DNA. You convert it to RNA. Step one, transcription. How do you do that? You follow the rule that A pairs with T and G pairs with C. A pairs with T and G, I'm sorry, A pairs with T and G pairs with C. Also, we know that in this case, T is actually U. So if you see on the bottom, all the words T, and let me maybe, T corresponds to A corresponds to U, C corresponds to G, G to C, and then A to U, T to A. So do you understand this part? So for anyone who had trouble with this, I probably did a mistake. Yes, I did a mistake. Whoever was that, very smart. Yes, thank you, Amibet. That was a mistake. That is UGA, that's not TGA. I cannot erase it, but I can definitely cross it out. Yeah, that's a UGA. Yeah. Yeah, thank you. So if you see that, so you are just writing the complementary base pair in this one, but in the second step of translation, the process is a little more difficult in the sense that you are going to use the 3-letter codon. It's called a codon, the one that translates to an amino acid. You take letter 1, that is A. I circled it. You'll take letter 2, that is U. So wherever the A and U meet, that column is the one to look. And it's AUG. So AUG. And whatever is in front, you just put it over here. Similarly, CUA. So that will be C with the U. So it has to be this one, CUA. That is L. And so you put an L over here. Okay. And in this manner, you can complete it. So at this time, I'm waiting for you to tell me the remaining amino acids up to all the way up to the end so that I will know that you understand this. Okay, so I think someone did do it, PEW. What happened to the last one? Whatever happened to UGA? What is that? Oh, people have different answers. Okay, that's going to be interesting. What is UGA? UGA is not C. What is written in front of UGA? Something different is written. All right, my friend, stop. Yes, that's where it stops. You cannot make endless proteins. The DNA is going to make, I mean, it's the cell is going to make a fixed amount of protein. So it comes to one point, then it should end. Okay. It should end there. So
don't put nothing where it says stop. Don't put anything.
You can put stop. You can put a stop code. Yeah, just write stop and that's fine.
Okay, act like you don't see my C. I put stop.
Oh, you did put stop. I see. Okay. Yes. So that's how you do it. And I want to see, wait a minute. So is there any questions on this particular transcription and translation part? Yes. Yes, people, I think you guys are getting it. I do think there is some controversy over TESPE. I see someone says TESPGW. So there is a little bit of contrary, TESPE. It's probably TSPE. So if you wrote TESPG, then probably there is some mistake. You probably need to go back and check what you're doing. Okay. It's not that hard. You can just check it, and I'm pretty sure you can find it. Okay, let me just clear this if you understood this, because I want to.
But why is the W right there? I don't
The
understand
W
why
is for tryptophan. I'm sorry. The W is for this one. I'm sorry. This is like,
Okay.
yeah, I'm sorry. That's what the problem is, I guess. That is what is clearly confusing people. Now, I do want to tell you what happens, but as we are telling, let me just, sometimes it's so hard to get out of these modes. Okay, so these are some of the functions of DNA. So what is these nucleotides are actually used for? Do they have a role, or it's just DNA, right? Okay, so oh, W was for both. Yeah, I'm so sorry, Lillian. Yes, you're right. And Gina, thank you for asking the question. That really was something confusing for people. So
No
W
problem.
is the one that is for tryptophan. Yes. So energy for metabolism, it provides energy. ATP. ATP is the energy currency of the cell. We all know that. So that is adenine triphosphate. So that's what one of the functions is. It serves in many places as cofactors. It helps in signal transduction. That's a mistake in spelling transduction. Those are the nucleotide functions, and nucleic acid help in storing the genetic information and passing it on, and help in processing that information. So these are some of the things. Now, since this one is going to be easier, before we switch on the topic to a little bit different area, I will have to show you something. Like, this is something, if you don't see, you will not realize. So let me just share something with you. I told you, like, HHMI Biointeractive has some amazing resources, guys, amazing. Okay, so what we are going to view here is transcription. It's going to be a very, I'm pretty sure it's going to be a really short video. It's like literally 2 minutes. But it's always so nice to be able to watch some of these things. I really, really enjoy this. I don't know how big this is. Okay. So yeah, let me just enlarge this and share sound. Also, for anyone who would want to watch it later, let me just share the link here. Control V, come on, you go with that long. Why it's not letting me share? Okay. Oh my God, I can't believe we're in the 40-minute endless cycle. Okay, so let me just share this sound with you and then start sharing this, okay?
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
Okay, so yeah, I think that was that was definitely a little too detailed, but it just gives you. I just wanted to give you a brief idea. There is not a whole lot of time left because of that 40-minute craziness, and I don't want it to get to us. So I do want to tell you that the slides after this are primarily focused on something which is much more easy to understand, and you can actually even watch the video. I'm just going to go over it very briefly. So the first thing that important for you to know is that DNA can get damaged. Even though the body has the polymerase, and polymerase is very careful, it's trying to replicate the DNA in the right way, but there is damage. You all probably have heard of sickle cell anemia. And so sickle cell anemia is a disease in which there's one nucleotide is different, okay? So difference in one nucleotide changes the amino acid, and that one change will make the hemoglobin sickle shaped instead of it being round, okay? I'm sorry, the red blood cell. So what it does is the red blood cells become sickle shaped because just the hemoglobin molecule becomes so sticky. So one mutation can have a dramatic effect in the entire body. It is something that we have to know. So if there is damage, first of all, there is lots of damage happening in every day. However, our body has a very nice DNA repair machinery, and it can definitely do a lot of repair, but not like 100% all the 100% time. And when there is unrepaired DNA damage, it can cause aging, it can cause a number of different diseases or different things. So that is the remaining part of the discussion. So how can this mutation happen? It can happen due to so many different reasons, because of chemical, because of radiation, because of UV, because of oxidation or any kind of errors in cell. All of those can lead to these different errors. For example, can lead to cell cycle arrest or DNA replication or inhibition. It can cause a lot of different things. It can even cause chromosomal damage, okay? And especially, you might have heard about the damage due to UV or radiation. So UV can cause the pyrimidines, the thymine, to kind of join among each other from the outside, which is not the right thing to happen. So when that happens, when you are outside and you don't have a sunblock on, or you prolonged exposure, it can definitely affect you because it causes the dimerization of pyrimidines. And if the damage is not fixed, then it can result in that cell it can possibly make that cell cancerous. And that is the reason for skin cancers. Similarly, ionizing radiations can do a very similar thing, okay? So all of these mutations can lead to adverse effect in health. Not like they won't cause you fever or cough or something, but they will change your cell, which in turn will change, your body in ways that you may or may not be even able to control. But so this is the UV damage that we're talking about. These thymine dimers, they're not supposed to connect like that. They're not supposed to be connecting here. There should be no connection here. However, they do connect. And if you see here, they distort the DNA. This is not how the DNA is supposed to be. They distort it, they cause a kink, and that causes that skin cell to become bad, right? And become a bad skin cell, which can even replicate and that replication causes skin lesion, and that is actually cancerous, right? So you might have heard of those tanning beds or whatever. All of those can cause that effect. Now, about the damage can be direct. The damage can be just the radiation can directly impact and break or do some kind of change in the structure. It can also be indirect. Sometimes when the high energy radiation is hitting the water, it can split up the water into free radicals. Those free radicals also have lots of energy and they can hit the DNA. So the DNA either is hit directly or it is hit indirectly with the help of water. So those are some of the different ways in which it can damage the DNA. However, not everything is really, really bad about this. There is radiation therapy, and what radiation therapy does is that it is trying to damage the DNA of bad cells, right? So if it is a localized tumor, you give radiation, those tumors really can't survive. Their DNA is sometimes like horribly damaged and they will die. Of course, the normal cells can also be affected, but just saying that this the radiation therapy or radiotherapy is ionizing radiation, which is put on DNA purposely of the bad cells so that they will die. Okay. So this is just like putting it really, really simple way. And just wanted to show you some pictures. So there is radiation therapy is used in various ways. And it has been used for a very long time for treating cancer. Okay. So about 2/3 of the cancer patients do receive radiation therapy as a part of the treatment. And like I said, the premise is damage the DNA of the cancer cell like really, really bad. And you do know that radiation therapy has side effects because it does normally also damage the regular cells. So there are different ways of giving the radiation. There is external beam, there is internal radiation, and there is proton beam therapy. Proton therapy is way more controlled and a little bit better that way. You can read about different things that how it is being used for treatment of tumors for curative purposes or for after resection to prevent the reoccurrence or for different purposes, right? And it can also be used, the radiation can also be used for x-rays or for CT scan or fluoroscopy. So this is something that you can read by yourself. There's medical x-rays, there is dental x-rays, all of these use radiation, and that is perfectly something that we use. I do know that we are running a little bit short on time in terms of what the clock is doing to us. If I do want to say one thing before we completely log out. I will be logging back in, that's one. So if you have any questions where I went fast, you can log back in. Second, if you have not given the oral part of the exam, please feel free to log in and do it now. Okay. So again, if you have not given the exam, you can do it now. So for the rest of the people, let's just continue to do what we are doing. Let's not fall behind. We have to finish this strong. So keep doing what you're doing and have a wonderful rest of the weekend. But I will be logging back in if you have any questions. Okay. And I promise to fix this by next time.